All right, so now that we've learned about artificial insemination, let's take a look at embryo transfer, which is going to involve primarily looking at the females as opposed to collecting semen from males. Embryo transfer is defined as taking a cluster of embryonic cells in a pregnant mother and transferring them into surrogate mothers. And again, similar to cloning, this is a great technology because we're able to help mothers or females that may not necessarily be able to reproduce or have reproductive complications and take their cells and put them into a surrogate mother where that offspring or that fetus can further develop until it's ready to be born. Some key benefits include rapid advancement of genetics from the mother, progeny testing, so we can figure out how valuable an animal is as a parent. It's safer than traditional breeding because we're just looking at the specific embryos and not mating actual animals like live in person. The use of dual production systems. So by using embryo transfer, for example, in the dairy industry, we can choose to have pure beef animals as our offspring by selecting the correct embryos. And it's, of course, easy to convert herd from grade animals to purebreds as a result of embryo transfer. And with all of that considered, it's pretty obvious that the big concern here is going to be that we're going to have a narrower genetic pool because we will be looking at specific traits and making them more pronounced. And especially looking back at that first one, rapid advancement of genetics from the mother, with super ovulation, which we'll talk about here in the next slide, we're able to get 12 to 15 eggs in one estrus cycle as opposed to just one. So think about this, if we're able to get 15 eggs or cells that are sent out after one ovulation, like that's going to be a lot more advancement of genetics than a simple breeding system would ever be. So before ET occurs, embryo transfer, we have to select the superior donors and recipients. We want to have the best of the best for our donors and our recipients. Again, donors is the animal that is donating or giving up their embryos, and the recipients are the ones that are receiving it. Donors and recipients are synchronized or put in sync, if you will, so that they are at the same phase of the estrus cycle. Superovulation then occurs, and this is when the donor animals release 12 to 15 eggs instead of just one during that estrus cycle. We then inject the donor or the animal that's going to be receiving the cells with the follicle stimulating hormone, or FSH. And this allows ovaries to produce several follicles, and this is where the eggs end up developing. 2 to 3 days after this occurs, we inject GnRH, or gonadotrophin, I can never pronounce that word, releasing hormone, GnRH, or a luteinizing hormone, which causes her to begin estrus. 48 hours later, the female then is in estrus, and this is also known as heat. When we are actually undergoing embryo transfer, fertilized eggs or embryos grow for 1 week and then are collected through flushing. The flushing procedure is as follows. We insert a thin rubber tube or catheter with the solution through the cervix and into the uterine horn. This solution causes embryos to get carried out of the uterus. An average of 6 embryos are collected with each flush. We then strain the embryos from the solution and examine under a microscope, and this helps us ensure the quality of our embryos. The embryos are then transferred directly to a recipient, either the recipient female, or they can be frozen. Finally, after embryo transfer occurs, the recipient cow comes into estrus using a feed additive containing progesterone. When the corpus luteum, or CL, reaches the correct stage, the embryo is placed in the uterus of the recipient. And it's very important during this process that we ensure that all animals and all equipment that's being used is very clean and sterile. Pregnancy progresses as it would in a normal conception, and then compared to natural conception, pregnancies from embryo transfer are just as likely to go full term and result in delivery of a calf or of an animal. So embryo transfer truly is comparable to a normal or natural conception that would occur in a traditional breeding program. The diagram over to the right is the corpus luteum, and I'm going to blow it up here in case you want to look a little bit more at this. I'm not going to expect you to memorize or know what all these different parts are because I didn't really talk about that. But if you are interested in this type of reproductive technology, embryo transfer, like I said, we talk about it a lot in the advanced veterinary science course and in the college and the schools course. So if you're signed up for one of those or you're looking into that, we will spend a lot more time talking about the CL and how that plays a role in reproductive technology. We are going to pause here, and the next video will focus on somatic cell nuclear transfer and a little bit of a more eloquent explanation of what Mr. Ramstead provided you here.